Hi, Kirko. Hi, everyone. Come up. How are you doing? Thank you for coming.
Morning. I literally just woke up.
that's good. That's even better. I mean, what then that you're coming, although you just woke up. So wait, let me make you moderator. So we will start in around 5 minutes. Thank you everyone for coming, and we will talk about honeybee research from Dr. Oshie Weller, and looking forward to see how people are trying to save the bees.
Hello, everyone.
Hi, Jamie. How are you?
I'm good, thank you. How are you, Katarina?
Good, good. How's your day so far?
Yes, so far so good. I was just rereading the honeybee paper, the bees paper, writing down some preemptive questions if we've got time for it. How about you? I think my day's like mostly over. Well, not almost, about halfway done. How are you? What's you
Right,
doing?
but you were up so late. That's amazing. Yeah, my day
But
was,
maybe you could do a paper on my brain one day, Katarina, eh?
yeah, seriously. I need more sleep than you.
Yeah, but you also work like 17 times harder than me, so.
I don't know.
Let's
Yeah,
see.
my day, I didn't, I went through emails and responded stuff, like drank some coffee, stuff like that.
That's
Didn't
your
do that
flaw.
much.
I drink tea exclusively. Coffee, that's that's your
I
downfall. Coffee, it'll destroy the world one day.
destroy the world.
Katerina, see when you put up a room and then it's finished and it's saved, you can't pin up anything new to it or anything, can you?
Add stuff to it once it's
Yeah,
done
yeah.
and
So you know how
recorded.
you pin it, you know how you pin up the paper for the speaker, right? And so when anybody listens to the replay, they could look at the paper. Could a speaker be there, do this talk, the room be closed, it's now a replay? Could someone open up, like, could we like open the replay and actually pin a new thing up? Like,
No,
no, it
no.
can't be done that way. Okay, right,
Mm.
that's good to know, because I had a couple of ideas which are being scrapped now because of that. Right,
Uh,
okay.
It's so cool that you're coming up with new ideas and doing them. It's great.
But you could, whatever you wanted to like change in the title, you could just put that like when you share it. You know what I'm saying?
Mm-hmm.
And give me share the room.
Do you mean like put like a new link or something in the title when it's being shared? Yeah.
Well, you can't put a link in the share, but I thought you were saying like change the title, like as like the presentation goes on or something. But I was saying like if that's what you're looking to do, you could always like do that with like sharing. Like if you share, you can put whatever you want to put in the title. There's not a link.
Yeah, yeah, no. I was talking about like the doctor here today. He's going to talk about the honeybees,
Hi,
and they're
Thomas.
going to. Oh, here we go.
Say hi.
Hello.
Hey,
Hello.
everyone.
Can you hear me okay?
Can you hear us?
Yep,
Can
I can hear you.
You're a little
I can
bit
hear you.
far away from the microphone, maybe, or
One sec.
maybe your headset is on low or something. We can hear you, but it's very, very like.
I'm just gonna try taking use a different microphone.
Cool.
So how about now? Is that better?
So much better.
Are you getting any reverb from anything you say? Because I've just taken the headphones out. So if you talk, maybe it will reverb. But
So far, so good. Testing. I'm not hearing anything. You, Katerina?
No, everything is perfect.
Okay,
Yep.
great.
Yeah, the Clubhouse. Jamie will laugh because I say this all the time. Clubhouse and really great, expensive headphones don't go together. I thought I would do something wonderful and buy new fancy headphones so there's less background noise when I'm in the lab, but it did quite the opposite. It made everything worse. So
Yeah, there might be a window that automatically closes in my office halfway through the talk. So if you hear that, I do apologize. But
I hope it was good so far.
yeah.
You
So
already met Jamie and meet Kiko and Serena. They are one of our moderators.
Hello. How are
Hi,
you?
Serena.
Good morning.
morning.
Pleasure speaking to you again, Doctor. Really looking forward to this talk. The paper is really interesting.
Yeah, well, I could talk about this more in a minute, but I gave some thought because I haven't ever really tried giving a presentation on audio only before. And so I was kind of debating how the best way to go about it would be. But I actually now think that maybe using a sort of more broad overview in terms of the talk, and then if there are specific aspects from the paper itself that people are interested in, I can then delve into those in detail, because I don't know whether everyone will have access to the paper in front of them while I'm talking.
Well, Katerina's pinned it up there, and so the people that will read along with you can read along with
Okay.
you. So, and that's actually a good idea though as well, because we have people of all different specialities here with us today as well. So, yeah, I really appreciate your approach. That sounds really good.
Okay. Well, one thing I will do is I can potentially reference figures that are in the paper if there's something relevant to what I'm talking about at that moment. But I don't want to make it dependent upon people being able to see those figures, if that makes sense.
That's actually perfect. Thank you. That's actually the perfect way you could do it.
Great. I think we can slowly start. Yeah, I think let's start the room. Welcome everyone to the Science Society. Happy Friday, everyone. And a special welcome to our guest speaker, Thomas O'Shea Weller. And he will talk about his research about honeybees and making them more resistant. So we will have honeybees in the future. It's a very important work. But first, before we start, let me give you a little bit information. Dr. Thomas Weller, he is a research fellow at the Environment and Sustainability Institute at the University of Exeter. And he did his bachelor in biosciences at the University of Exeter, and his PhD at the University of Bristol in biology. And he is interested in complex interactions that govern collective behavior, ecology, and self-organization within social insects. And his research is focused on the intersection between fundamental investigations of colony function and applied work in ecology and epidemiology. And he works with ants, honeybees, bumblebees as models to assess trends of ecological and organizational concern. He's involved in different research projects around that. And welcome, to the Science Society, Thomas. And if it's okay, Jamie will ask you first a few questions, and then we go ahead to, the stage is yours for presenting your research here. Thank you.
Great.
Cool. So, first of all, officially, thank you very much again, Doctor, for coming and joining us. Your bio is really exciting and impressive, and you must have so many stories to tell. But first of all, I'd like to ask, what is it that first got you into science in the first place and get into these kind of fields before we delve into the bee stuff specifically? When did you first decide that science was your calling, and how did you get there?
Yeah, that's quite an easy one, actually. When I was a little kid at school, I was always super interested in ants, and I like to dig up paving stones and various other things to see what the ants were doing. And at the time, I mean, it was just playing around because I thought it was just fun to watch them, going and putting a little bit of sugar solution or some jam and seeing how they'd congregate around it. But I never really grew out of that habit, I suppose you could say. And so what it I think was that fascinated me so much was the kind of complexity in their organization and how that's in many cases quite similar to some of the things that human societies do.
That's actually cool. And so is this what led you into then focusing on the bees and your current paper and your current problem that you're working on? What got you into that specifically?
Well, more or less, I guess. If I'm honest, a lot of the sort of carryover into honeybee research is because honeybees are really, I suppose, the most economically important for the most part in terms of social insects, because obviously many human systems and agriculture and pollination are quite dependent on bees. So a lot of my research focus had shifted more towards honeybees over time, just because there's demand and application. Whereas working more with bumblebees, some of the wild species that we have in the UK, and working with ants, that's always been more of a sort of theoretical interest aspect. So work done there might not have some direct applied use, but honeybee work is always quite useful from an applied sense.
That's really interesting because I've heard of ants being looked at for behavior and inspiring a lot of computer architecture and behavior and stuff like that. So that's interesting that bumblebees are what you're looking at from a biological perspective. Right. Well, thank you very much for that. That's always appreciated that you would share that with us. And when you're ready, the stage is yours, and please begin.
Okay, great. Right, I will start from the beginning, I suppose. What I'm going to try and do in this talk is keep it nice and concise and short, and also aim more for a sort of higher level overview of the work. And then if anyone has very specific questions that relate to things in the paper, then we can focus in on those at the end. So, where this all really began and the impetus for this work stretches back to the commercial pollination industry in the United States. We have something known as migratory pollination, which is where colonies of bees are moved around the country to provide pollination services for different crops as they come into bloom. The process is sort of often known as chasing the bloom, and it's incredibly important in terms of providing pollination services. And in fact, some crops, like for example, almonds in California, they're obligately dependent on honeybees being brought in to do this because there aren't any natural pollinators in the local environment. It's very big business, but it's also an incredibly stressful system for bees to be in. And we can see this reflected in the fact that there's very high mortality on a yearly basis. There's a lot of various figures that you can kind of throw around relating to this, but I just have in front of me right now some values from the last 2 years of overwinter mortality for bees in migratory operations. And they're coming in at 22.2% for a year ago and 32.2% for last year. And that's just over the winter. So it's quite a dire situation in that it's not very sustainable and also ends up costing a lot of money both for beekeepers to be able to maintain their colonies, and also for other workers involved in the system because of all the support infrastructure that you need around the system just to keep the bee colonies alive. Now, in terms of why exactly this happens or why mortality levels tend to be so high, it's a multifactorial problem, as many things are in biology, but there are a few sort of prospective key culprits that often emerge from the scientific literature. And just to summarize very quickly some of those, we have limited nutrition and forage. Because bees in pollination operations are in agricultural environments, they often only have access to monocultures of different crops. And it's been shown to an extent that without the dietary diversity of pollen that they're used to, it can do various things that weaken their health and increase susceptibility to other factors that may harm their health. We then have pathogens and parasites. Both of these are a particular problem for large commercial operations because the colonies are often in very close proximity to each other. So as you can imagine, a bit like if we have battery hens or industrial farming of any animal, the chance for disease and parasite transmission is greatly increased with concomitant effects on mortality. Finally, we have two more human-mediated aspects. There's agrochemical exposure. So in agricultural systems, you have fungicides, pesticides, and many other types of chemicals that the bees are likely or more likely than usual to be exposed to because they're constantly in these systems. And finally, management stresses, because colonies are moved all around the United States and Canada on the back of trucks, and also moved from one climate zone to another in a way that they would not really ever experience naturally. And so this also can take a toll. So I've talked about these different problems that are faced by bees, but today and in the work that I'm describing, we're really focusing on one particular issue, which is Varroa mites. Varroa mites, or to use their full name, Varroa-destructor, which I think is a great name and it's pretty indicative of what they do, are really a focus of a lot of the breeding work that occurs in honeybees because they're widely known to be the largest threat to colony health. A nice little fact that I often like to come up with to describe this is that almost all of the colonies in both commercial operations and just casual operations in the United States and Europe are likely to be infested by Varroa mites to some level. And in many cases, if they're not constantly treated with miticides to keep the mite levels under control, the mites will kill them, which is a very sort of bleak reality when you think about it. And so what we are very interested in from a research perspective is developing honeybees that are able to resist or at least control the population of these invasive mites. And the reason that we think breeding is a great idea comes from a lot of different aspects. But one interesting thing is that Varroa mites are an invasive species for European honeybees because their original host is the Asian honeybee, Apis cerana, which they are quite well adapted with. But when they hopped species to the European honeybee, Apis mellifera, the bees really didn't know how to respond to them and seemed to be largely oblivious to these mites, so they can wreak havoc within the colonies. However, some of the behaviors that the Asian honeybees have to deal with mites are also present in low levels in European honeybees. So there's an ongoing demand to try and select for these sorts of phenotypes and breed it in to existing stocks of bees to provide resistance. Now, the methods by which bees can be resistant to mites are quite varied and numerous. We have things like Varroa-sensitive hygiene, which is in large part what we're going to be talking about today. This is a process where the bees can detect infested brood, and they'll uncap the cells and then remove the infested brood and throw them out with the mites in the cells. And that's very effective because the mites reproduce in the brood cells, and all of the progeny mites and the male will die if the cell is opened. They can't survive outside. Only the female can survive outside. So by uncapping cells and removing the brood, they bees are able to stop the mite's reproductive cycle, and then they can never really reach these exponential levels that cause colonies to die. And there are various other adaptations, like bees that will attack the mites out in the open and bite their legs off, which is often known as the mite biter phenotype of bees, whereas other behaviors such as swarming are also useful for keeping mite levels low. However, we find a sort of paradox that occurs with all of these breeding approaches, is that it's not a zero-sum game, in that by selecting for particular characteristics to make them resistant to Varroa, we often lose the characteristics that we need to make them useful for beekeeping. For example, colonies that swarm frequently are not very good from a beekeeper's perspective because they're almost untenable in terms of management. They're swarming the whole time. Bees that, for example, bite or remove the legs of Varroa, they don't necessarily have large colony sizes and don't necessarily produce a lot of honey. So again, it sort of defeats the object to an extent if you're losing the other characteristics that are desirable in the pursuit of mite resistance. So we have that ongoing issue, and that's where the paper that I'm talking about here comes in. We have basically been part of a decade-long, or more than a decade, breeding program to come up with a variety of bees that is both very resistant to Varroa and also ideal from a beekeeping perspective in terms of those characteristics. And these bees are known as Pol-line bees, which is a somewhat cheesy play on words from pol as in pollinators and line as in genetic line. And they've been bred in an interesting way, which is we've taken the VSH, or Varroa-sensitive hygiene behavior, this behavior of removing infested brood. That's been bred very strongly into the bees, and then they're outcrossed with existing standard commercial strains of bees that have really large colony sizes, honey production, and are nice and docile. And so what you get at the end of that process through repeated year after year cycles of selection is bees that combine the best of both. So they're ideally resistant to Varroa, but also able to produce nice large amounts of honey, are very docile and nice to work with. And finally, colony sizes are large, which makes them great for pollination. So I think I actually have one of the materials provided with this talk that shows a PCA analysis. I'm just going to see in the materials provided what is labeled as. So for anyone who has the paper up, the very last page of the provided material is there's an additional figure 1 and an additional figure 2. Additional figure 1 shows a genetic PCA analysis of different stocks of bee. And it's quite interesting because it shows that Pol-line bees and a quite closely related sort of sister progeny stock called helo bees are very distinct from all the other stocks of bees that are currently being developed for mite resistance. And the reason that that's somewhat important is because it shows that through this intensive breeding, we've been able to really produce stocks that are incredibly distinct and not necessarily going to lose the characteristics that they have quickly through outcrossing. So it's a moderately robust outcome, which I think is one of the key goals. Right, so we've talked about Pol-line bees and how we bred them really to be good and have the best of both worlds in terms of both beekeeping characteristics and also mite resistance. But it's all very well and good to say that. We really need to validate these to make sure they actually do what they were made to do in real, commercial operations and in real scenarios where they might be used. And that's where this study comes in that we were aiming for a large scale nationwide in the US experiment where we put the bees into varying different climate regions, because just because something performs well in one locality, it doesn't mean it's going to perform well everywhere. We replicated the experiment with a large number of colonies, several hundred, and we took very detailed measurements across the course of a year to find out for colonies that died, why they died, and for colonies that survived, why they survived. So if we look at figure 1 in the paper, And for anyone who doesn't have the paper in front of them, figure 1 is a map of the United States showing where the bees were kept and where they were moved. And for the general experimental layout, we had half of our colonies were the mite-resistant Pol-line bees, the ones we wanted to test, and the other half were a standard commercial variety that's sort of the best currently available bee that's used for the most part in pollination operations. So we really wanted to see, if we test the best currently available type against what we've produced, do we get any added benefit? And the colonies that we have, we conducted various manipulations to test different aspects. First thing we did was, for all of the bees we had in both stocks, half of them received a normal treatment for mites, so a miticide treatment for Varroa, which is what would usually occur. So standard mite treatment to keep mite levels low. The other half received this sort of minimal reduced treatment, which really would never usually occur. But the reason we had this other group is because we wanted to see, well, if we don't really treat them for mites properly, can they still survive due to their own ability to be Varroa-resistant, and how does that compare to the current commercial varieties that are used? Finally, we had 3 different migration routes that we used just to look at the effect of different scenarios in terms of crops that they were put among, and also in terms of climate. One of the migration routes was bees that were kept in Louisiana, Mississippi, moved up to South Dakota for honey production to see how well they produce honey, and then overwintered by being moved back down to the start point. The other group went up to South Dakota, as with the first group. Then they went to California to overwinter and provide almond pollination in the spring before returning back to Louisiana and Mississippi at the end of the experiment. And the final group remained stationary. So they were, for the most part, in Mississippi for the whole experiment and not moved at all. So that sets the scene for how we went about devising and setting up a large-scale experiment with the real aims of saying, how well do Pol-line bees survive compared to standard stocks? How much honey do they produce? Do they produce enough honey? Is it comparable? How large are their colony sizes, especially in California? We want to be able to pollinate the almonds. And then finally, if we don't treat for mites properly, if we let mite levels get high, what is the result in terms of how Pol-line bees are able to perform them? So moving straight into some of these results, I think you can draw your attention, for anyone with the paper in front of them, to figures 2 and especially figure 3, which are really showing the survival rate of the 2 stocks and also the levels of Varroa in the 2 stocks. And what we see is that in, I think this is figure 3A and B, commercial versus Pol-line Varroa levels, is that throughout the year, the Varroa levels started to increase in both stocks. And then in the commercial colonies, by September, levels of the mite infestation had gone absolutely sort of stratospheric and completely out of control, which resulted in killing a very, very high proportion of those colonies. However, for the Pol-line colonies, during the same period, very few colonies ended up with high mite levels. And in fact, they were able to survive a lot better and kept that Varroa under control. And this was even worse when considering the colonies that weren't fully treated. It was pretty much a guaranteed death sentence for commercial colonies, not so for Pol-line colonies. And the overall outcome of this, when we just looked at both of the stocks and compared survival rates, was that the commercial colonies had very poor survival, but the Pol-line colonies had roughly double the level of survival. So there were double the number of Pol-line colonies alive at the end of the experiment compared to the commercial stock, which is great from our perspective because this is really the worst scenario that we could put them through. Then looking at honey production, which I think is to an extent covered in figure 2, but again, for anyone without the figures, we found that honey production was equivalent between the 2 stocks and colony size was also roughly the same. But in reality, because far more of the Pol-line colonies survived, they did produce more honey and had larger colony sizes overall. So that kind of is interesting from looking at the survival perspective, the health and Varroa levels, but we also conducted some analyses of some of the pathogens, specifically viruses, and the levels present in the two stocks throughout the experiment. That is, I believe, mostly covered by figure 3, C, and D. But what we found was that when looking at 4 major honeybee viruses, we had black queen cell virus, chronic bee paralysis virus, and deformed wing virus A and B variants. What we found was that deformed wing virus A and B, there were broadly reduced levels in Pol-line colonies, which makes sense because these are Varroa transmitted viruses. So we would expect lower levels of Varroa means lower levels of the viruses. For chronic bee paralysis virus, there was really less of a trend here, which again makes sense because chronic bee paralysis virus is potentially somewhat transmitted by Varroa, but it's not the main pathway. Whereas for black queen cell virus, which is not transmitted by Varroa, there was really no difference between the two stocks. And that kind of begs the question then is why, or exactly what do we think is going on with these Pol-line colonies having much lower viral levels? And it appears that it's nothing to do with resistance to viruses. If for anyone who has the document with them, additional figure number 2 shows some comparative viral titer estimates for when bees of each stock were injected in the lab directly with viruses. We see that really Pol-line bees aren't any more resistant to the viruses themselves than are Italian or really any other stock. And so really what's actually happening is that the reduced virus levels are due to reduced Varroa-mediated transmission due to lower mite levels. So that worked out quite nicely in terms of being able to see where the mechanism that was occurring here was how was it working. But on top of that, in terms of the reason for lower Varroa levels really improving the health of colonies, it's not just the viruses that are transmitted by Varroa mites, but it's the act of feeding itself that does a lot of the damage. And for a long time, it was thought that Varroa mites fed on the hemolymph of bees, which is their blood effectively. But some recent work showed that this actually wasn't the case, and that Varroa mites mainly feed on the lipid body, the fat body of bees. And this is pretty significant because the fat body of a bee is quite analogous to sort of the liver of a human. It acts as a detoxification role. And so if a Varroa mite, and bear in mind that compared to the size of the bee, the mite is almost the size of the bee's head, so it's a massive parasite, begins feeding on the fat body of the bee, it's doing a lot of physical damage to that bee and also making it vulnerable to any other processes that may affect toxicity. So we see that feeding alone does a lot of damage in and of itself. And I think one of the interesting takeaways from that particular result is that when we're thinking about ways to deal with both viruses and with Varroa, we should often really think about focusing on the Varroa because it's the upstream factor, because it does damage, but it also transmits viruses. So if we just focus on viruses, even if we can completely remove or find a cure or a treatment to a particular virus, it's not going to solve the problem with the Varroa doing a lot of damage by feeding. However, if we take Varroa out of the picture, we stop that direct route of damage by feeding, and we also, as a corollary, remove all the viral transmission that's conferred by that. So one final, I think, interesting takeaway from the particular piece of research that we were surprised to find somewhat was that when we analyzed the relative importance of Varroa versus viruses, it seemed that viruses actually most of the time, or the vectored viruses, weren't very good at predicting whether a colony survived or died. It didn't seem that the level of virus was that indicative of what happened to the colony, whereas the level of Varroa was. And we were able to look at this because we could select out colonies that had the same level of Varroa but differing levels of virus and vice versa. And the reason that's quite interesting is because it's not something that was widely thought to be the case in the literature before. I think often Varroa mites are kind of seen as primarily a vector of viruses, and that's how they do the damage, a bit like, a mosquito's may kill people through transmitting malaria. It's not the mosquitoes feeding itself that does the damage. However, with Varroa, I think actually it might be much more shifted in the opposite way, whereas the viruses are sort of a secondary effect, and the feeding itself is what is causing a lot of damage, which again is a useful thing to know because, like I mentioned earlier, when we're thinking about ways to practically deal with Varroa mites, it's good to know that we should aim for the mite rather than necessarily the pathogens that come as a secondary factor. So overall, we're quite pleased with the results because it provides at least some evidence initially for a sort of sustainable solution to Varroa mites. But the key thing really is that we have current treatments that we use against the mites, amitraz and to a lesser extent, I believe, Coumaphos, two products that are used as miticides, but the mites are developing resistance to these products. So it's something of a race against time, really, because we're not coming up with any new chemicals to use against them. And as mite resistance levels to these miticides increase over time, they're becoming less and less effective and having to be used more and more. So having some other mechanism by which bees can be resistant is incredibly important. And in fact, for Varroa mites, they can often evolve quite rapidly against chemical mechanisms of harm because they have high levels of inbreeding and rapid population cycle time. So if there's a change in a potassium channel gate or some such that only requires a small level of mutation, then it will likely occur and proliferate. However, becoming resistant to the behavior of the bees that are detecting and removing the mites, that's a lot harder, and that's something that can't evolve nearly as quickly. So it's more sustainable in many cases to let the bees kind of do the job for you rather than needing to apply more chemicals. And then finally, along with that, it's, I think, more environmentally sustainable because you're just having to reduce the amount of beekeeper intervention and the amount of products that you're using on your bees and that are making their way into the environment. Okay, so to sum up where I think future research will be going now, we've learned quite a lot from this first piece of work, but we have another year of data that we're currently working on for a second year of this study. And we're really interested in providing better prediction models in terms of being able to predict risk factors and when you need to intervene to deal with these risk factors. So like level of treatment for Varroa mites, whether certain pathogens or parasites, at what level or at what situation do you need to be worried about those and really try and act and treat your colonies. And also what weather patterns and management regimes are good or bad, and taking that from a sort of probability distribution approach rather than just binary yes, no results. And on that note, I could probably draw your attention to the figure 6, I think it is the final figure in the paper. That's just a nice little scale looking at levels of Varroa and colony outcome. And the reason that's slightly interesting is because currently when it comes to treating Varroa mites, often there's a rule of thumb that you should treat for your mites if you have an infestation level of 3 mites per 100 bees. And we really were kind of finding that that is not conservative enough. Really, by then, a lot of damage is already done. And I think this is something that requires further investigation in terms of telling at what point you actually need to take control measures and deal with some of the emerging risk factors. Because when it comes to mites and other parasites and pathogens, the growth and the way these grow in the colony is often sort of exponent, where once they reach a certain level, they just completely take off and are likely to do a lot of damage very, very quickly. So a final note, this isn't particularly covered in the paper, but I thought it would be a nice add-on at the end, is that having had this and additional years since this work was done of successful tests for Pol-line bees, we have started to really roll out a program to make them available, or the queens from this genetic line available to beekeepers and bee breeders, and that is the Helo bee program. So Helo bees are a derivative of Pol-line. Really, the only difference is that they're being bred in a program in Hawaii to make as many queens available on a yearly basis as possible, and then to distribute those at least at a preliminary level, to beekeepers. And there's a pretty nice website that they have set up at the moment, helobees.com, where you can see more information on that. But the takeaway I think that's really nice here is that helo bees and Pol-line bees, they're not just bees that can be used by commercial beekeepers and by large operations, but even just hobbyists who want to keep bees, they're ideal because they require a lot less input and management than many other stocks of bees, because they can be resistant to any varroa that's coming in the local environment, and they take care of themselves, and they're also nice and gentle. So I recommend them. I like working with them for that reason. So yeah, that's kind of where we're going at the moment. Is just we've had some quite nice successful tests, and we're really just interested in providing some more tools, but also rolling out some of these resistant bees to beekeepers. So I'd probably just like to wrap up by thanking all of my collaborators, especially at the USDA Honeybee Breeding and Genetics Laboratory in Louisiana, LSU, the university that we also collaborated with, and finally the University of Exeter, where I'm based now, and all of the other field personnel and colleagues in the US without whom this work would not have been possible. So I think that brings, for the most part, my talk to an end, and I'm happy to take any questions you have and have any further discussion that you might be interested in.
Thank you so much, Thomas, for this amazing presentation and this very important and interesting research. First of all, I'm not sure if I'm the only one, but I was so amazed about the span of migration. Like, how long do they take for, I'm sorry to ask this, I know we're mostly focusing on the resistance and fighting of Varroa, but how long do they take for these migration cycles?
Yeah, so in terms of there's two aspects. There's the travel time, and then there's how long they spend in various areas. And it does vary from operation to operation, but it's pretty extensive. So on the back of a truck, it can take several days to move bees from one location, several thousand kilometers, several thousand miles, up to another one. And then once they're there, they usually spend a few months in these different environments. So in almonds in the spring, they'll usually be moved out for most of the winter, and then through the whole of the spring when the almonds are in bloom, where they provide pollination, so several months. And then once the almonds finish blooming, they'll be moved back to wherever they were originally based. In the Dakotas, I mean, for example, I think we kept 3 or 4 months in South Dakota, just so that they can really start foraging in the local vegetation that's available there, and various crops, and build up a large amount of honey. Then that honey is harvested, and after that's done, they're usually moved, because South Dakota gets, as you can imagine, quite cold in the winter. So yeah, it's a multi-day process, quite often just in the transit time.
So does Varroa vary? I'm sorry about being so ignorant in this field, but when, let's say you have kids, right, and you move to another school, kids get constantly sick like the first couple of months because the cold viruses and things like that slightly vary. So is that also a contributing factor of moving them around and having maybe slightly different types of varroa?
So in terms of the sort of effect you get with children, where they pick up a different strain of cold or flu, I wouldn't say that is the case with Varroa, because all bees, or all European honeybees, are super susceptible to Varroa. Varroa is kind of like a zoonosis, so it's going to infest and destroy colonies regardless of where you get it from. But the second part of that is the point that in different regions there could be different sort of endemic levels of Varroa. So let's say you moved your bees to an apiary in one region, where there were other apiaries and people had completely infested colonies. So maybe a better analogy would be head lice. You know, if you send your children to a new school and everyone's riddled with head lice, then they're probably going to get head lice as well and be exposed to it. So there are these sort of potentially dangerous situations where they're moved into areas with higher levels of infestation, and that can be a real problem.
I have a question here, Thomas. What are the precautions that I should be taking? Like, you have these honeybee homes, right, or whatever that you call them. So, as an individual, what are the precautions that I should be taking?
You mean the precautions in terms of Varroa treatment or dealing with the potential threat of Varroa? I would say there's two recommendations that I can give, and I mean, depending, you may already do this, but the first thing is testing your bees to see how infested they are, and that's, I mean, one way. Well, there's multiple ways to do this, but popular methods include having a board under the colony, and then mites, or a kind of sticky board, and mites that fall off the bees will drop down onto that board, and then at a set amount of time, say a day or two, you count the number on the board, and then there's a lot of literature available and a lot of guides available to say, you know, how much is a problem or how much is too much. The other method, probably I would say a preferred method, is that you take a sample of bees from your colony, and then you can cover them in powdered sugar and shake them, and that will make the mites come off of those bees. And then you can, again, count the number of mites you're getting. And as long as your sample size is roughly equal each time, so if you use a container of the same size, you can tell what level of Varroa you have. And if it gets too high, using a treatment such as amitraz, Coumaphos, or a number of other options, quite a few options available to control mite levels. And there's a lot of literature available on guides on the internet, and also beekeeping societies will have information on that can help.
Can I ask a quick follow-up question on this? Thank you, Thomas, and thank you, Katerina, and other people in this club. When you take the sample size and then you cover with the powdered sugar and the mites come off, do you lose those bees as well?
Thomas, you're muted. I'm not sure if you are maybe talking.
Sorry.
Yeah, just
Sorry.
Thank you. Thanks.
Call off us.
Yeah, so we didn't hear anything, Thomas. Thank you.
So to answer your question, it's a very good question, and that's actually why I would recommend the powdered sugar method, because it won't kill the bees. If you do that, once you've sort of shaken them up and got a mite count off of that, you can return them to the colony, and the other bees will usually lick them clean. There are other lethal methods which are quite commonly used. So some methods include soaking the bees in soapy water so that the mites fall off, but of course that kills the bees, or using various other solvents like alcohol. But if you're not aiming to kill a few hundred bees every time you sample, which you don't really want to do, especially if you don't have very large colonies, the powdered sugar method is preferable in that regard.
Hi, doctor. I actually live in the Central Valley of California near the almond farm. And I'm not a farmer, but I hear those guys talk about the colony collapse disorder. Is this parasite one of the causes of the colony collapse, or is that a different problem?
Yeah, that's an interesting question. Colony collapse disorder is quite a specific phenomenon, which I would argue is quite rare, but because it's called colony collapse disorder, it often gets confused and misreported. Colony collapse disorder refers to a spate of cases that appeared a few years ago where the colony appeared to be completely dead or absent a queen, and there were just a few workers left hanging around. And I don't believe that it was ever really pinned down what caused that. But the interesting fact about it is that most of the colonies that die, or you could say that collapse, we know why they die, and it's because of Varroa mites, starvation, or pathogens. And so, really, in many cases, it's possible that what was reported as colony collapse was caused by Varroa mites. But the interesting thing about colony collapse disorder as a phenomenon was it was kind of mysterious and unaccounted for. So I don't know whether that completely answers your question, but long story short, most of the collapses of colonies that occur, most colony deaths are due either directly or indirectly to Varroa or other known factors.
Yeah, sometimes I feel that the beekeepers are just hiking up their prices, blaming colony. Well, the years ago, so I know, some of these farmers, so they rent the beehives, right? So years ago, it's like $50 per beehive, and now it's like 200 something per beehive, and actually has gotten to be a problem because those beehives are now getting stolen. So they actually put like GPS trackers and surveillance cameras to make sure that those don't get stolen. And that's just an aside.
Yeah, quite an interesting phenomenon. We also had to kind of deal with this side of things because the operation we were working with, they have a lot of colonies and they're kind of prime game for people to nick and steal them just because they're all in one place quite often. So we came up with various inventive solutions to stop that from happening. But yeah, it's very big business. And a final note on that is that the price of a colony is very much determined by the size of the colony. So beekeepers make an absolute premium on very large colonies with a lot of bees in. And so that's often what they're interested in having as an end product. They want stocks of bees that produce really big colonies.
Yeah,
Um,
thanks, doctor.
I love the GPS tracker idea. I had a very general question, and thank you so much for this talk. Would you have any advice for the hobbyist just wanting to get started in, say, the Central Florida region?
Yeah, one thing I would say is if you're really starting out, try first of all to locate other beekeepers, especially other hobbyists or societies, and talk to them about getting particular queens or starting up from scratch. Because one sort of mistake that I think is often made by beginning as a hobbyist is that there are a lot of sources available to buy queen bees from. And I wouldn't quite go as far as to say that they're sort of relying on people not knowing what they're doing, but you can end up buying queens from quite poor sources that will invariably end up with bad outcomes, like the don't produce enough brood and the colonies die, or they're very aggressive, etc., etc. So invariably, beekeeping societies and local, I'm sure in Florida there's plenty. They'll already have a lot of information on what bees do well in our local environment, what reputable breeders are there, where do we get ours from, what can we recommend, and that is very invaluable. I mean, even if you're just shooting them an email or something and saying, I'm starting out, do you have any recommendations on where I get bees from, etc. The other thing I would say is try to start manageable and small. So 1 or 2 colonies. We've had a few people who get a little bit over enthusiastic and end up taking things on more than they can manage. And it's like, a dog is not just for Christmas, it's for life. Well, a bee colony is often potentially a lot more dangerous than a dog and they require a lot more expertise. So starting small and being informed by local beekeepers is really useful, I think.
I have a
Well, thank
question,
you.
Tom. I have a question here.
can I would like to read out a question from the chat first, please.
Sure, go on, sure.
Krishna, she asked, Thomas, can the mites be led somewhere else? Like, can there be a source of what the nodes want? So the one of the bees go to the source to access what they want more easily, Piper style. I hope I read the question right, Krishna. Do you see the question, Thomas, in the chat? You're still on mute, in case.
Sorry, I'm just looking to see the chat if I can find it.
It's all the way on the left hand. There's like 13 and a little bubble, speech bubble, on the bottom of your screen. Now it's 14. Can you see that? I'm not sure. You're on the desktop app, so.
I did, I actually can't see, yeah, because the desktop's different, but I just realized what the question was asking while I was thinking about it.
Okay.
So it was, can
My.
mites be led away from the majority of the bees in a sort of Pied Piper fashion by something that attracts them or another bee? I don't know, because I don't think that that's ever been attempted. But what I would say is that the mites are attracted the most to drone brood, because drone bees have a much longer maturation period in the cell. And so the mites really like to infest their particular cells because it gives more time to reproduce. So there is one method that is sometimes used called sacrificial brood, where they take drone brood, or the beekeeper takes drone brood and sort of lets all the mites infest the drone brood and then takes that out the colony and freezes it to kill the mites. But of course, that also kills a bunch of developing drones. So I guess that's the closest thing that exists to that kind of methodology.
Yeah, great. Thank you, Father.
Yeah.
Can I go ahead and ask a question?
You let me
Hello.
just yes, Varun. Can I ask, do you by any chance work with Glenn Jeffrey from UCL, University College London? He works on this deep red light exposure in humans, but he was a guest speaker here a while ago, and he also talked that he was working with bee colonies to like improve their health. And do you know by any chance his work? A deep red light basically improves, I guess, immune system and rejuvenation. So basically, you're healthier if you get exposed like for 2 minutes a day or something.
I have to admit I'm not familiar with Ben's work. I'll definitely look him up now. I guess one thing we do know, well, I say no, one thing that is interesting about bees, and I don't know whether this is deep red light, but when it comes to red light, a lot of the experiments we do where we want to observe bees without disturbing them too much, we use red filtered observation glass or panels, because it seems that they respond a lot less to light coming through a red filter than they do to standard daylight. It's in the case of ants, for example, it's nearly invisible to them for many species, and for bees, it seems that it might not be invisible to them, but it's definitely less disturbing to them. So I guess that's some overlap there. But I'll check out his work and see what I can find there.
Yeah, apparently, in humans, he started, it improves like the eyesight in elderly people. So apparently the mitochondria get kind of rejuvenated by deep red light. And it's only like a minute and a half a day in the morning. It only works when you do it in the morning. Apparently mitochondria have also circadian rhythm, and we don't know exactly why. And then he kind of, I don't know how he came about it to apply this to honeybees, and I think he's working in France with some farmers there, and it seems to be improving their health. So yeah, it would be interesting. Thank you. And Varun, please go ahead. But I wanted to check if Kirko, Christopher, if you have first a question, because they didn't get the chance to ask yet, and Jamie.
Thank you, Katharina, for giving me this opportunity to ask this question. So kind of you. Anyways, now how do I know whether the bee is the queen bee or not? Like, are there any physical attributes involved?
You mean in terms of identifying the queen bee?
Yeah, yeah, yeah.
Okay, yeah, that's quite easy. So queen bees are larger than workers, and their gaster, so the end part of the bee, is elongated because they have very large ovaries to produce all the eggs. But what is normally done among beekeepers is that once they find the queen bee in a colony, they mark her with a dot of coloured paint. So that way, the next time they want to look in the colony, they can check out and find the queen very easily to make sure she's in good health.
Oh, okay. One more question here. Like,
Sorry, let's move on to Kirko. I want to make sure that everyone gets a chance to ask here, and Jamie and Christopher. Thank you.
Yeah, so I was like kind of curious. When you were talking about like the like using like the Asian bees that are resistant to the Varroa destructor. Uh, so I was kind of like interested or curious, like if you do that, does that not dilute like because like half of the genes that the offspring would gain will of course be from um the Asian bees. Does that not change the viability of the bees that you're crossing that bee strain into?
So yeah, I think I understand the question. So a quick point: we're not crossing European honeybees with Asian honeybees to make Varroa-resistant bees. What we're actually doing is we're looking to see if some of the behaviors that Asian honeybees use to deal with Varroa, whether those behaviors are also present in European honeybees to an extent, and then we're trying to amplify those through selective breeding just of the European honeybees. So it's a bit like when it comes to, for example, species of, or even just stocks of cats or dogs or varieties, you'll find that there's a lot of overlap in things that they do and behaviors and characteristics. But some of them may be expressing these things to a higher level than others, and that's the difference. So you can try and amplify any given particular trait within a single species or stock, and then get that to a point where it's expressed almost as much as it would be in the other species, but you haven't had to cross any of them in terms of the actual Asian versus European honeybees.
Sweet, thank you.
And then just a quick question for you. Where do they actually pick the mites up from? I mean, is it environmental from the flowers they're pollinating, or do they just spontaneously come to the hives?
It's a bit of both. The mites can sort of hop off of a foraging honeybee onto a flower. There was some quite interesting work where they showed that sometimes mites will wait at a flower, and then when another forager comes from a different colony, they'll jump on her and she'll take her back to the hive, and then the whole thing kind of starts again. But that's probably not the main way that they end up being spread around. We think probably one of the key ways is through something known as worker drifting. So if you're a beekeeper and you have an apiary with a lot of colonies in, and there's workers flying in and out and foraging, sometimes a worker will accidentally go back to the wrong colony and they'll accept her. But if one colony is very infested, it will end up with a sort of bit of a transfer of some bees accidentally going to other colonies, and then they get infested as well. And then finally, on top of that, we have another issue, which is that some practices in the beekeeping industry will necessarily result in spread of disease. So, for example, there's practices involving moving frames of brood or other things from one colony to another to boost their health. Well, if they're infested, as you can imagine, it's a bit like getting a contaminated transplant in that you're then going to get whatever the contamination is, and in this case, it's varroa mites. And finally, this is really quite an extreme practice, but in some cases, colonies are made by taking a kind of bunch of workers from many different host colonies, throwing them all together, and then adding a new queen so that it will establish a new colony. And as you can imagine, doing that again, there's a high risk if the colonies, the source colonies, are already infested, that it just kind of plants the seed for more infestation.
Thank you.
Thomas, like when in new colonies.
Jamie, did you have a question? I saw you on mic.
Oh, I did, but if he wants to ask really first, I'm going to be I'll blast through my questions as fast as I can. But if he wants to go first, I don't mind. Quick.
Thomas, do you still have some time, or do you have to leave? Because it's about an hour that we went about. So, do you guys still have time for a few more questions? You're on mute. So,
Sorry.
no worries.
Yes, I can take a few more questions.
I have one when you're finished in queue.
Yeah. So maybe Jamie, Z, and Krishna, if that's okay.
Okay, great. This is actually an amazing talk, Doctor. I've actually got a whole bunch of questions, but I'm going to try and blast through as many as I can, quickly as I can. One of the questions I'm going to, suppose a three-parter. I'm wondering, what is the behavior of the mites? Do they behave like bees? Do they have colonies of their own? Do they have bees? Do they have like queens or whatever their own? Is there any way that that behavior of the mites itself could be monitored and exploited? And second part here is, could there be anything, since they like the fat, could there be anything given to the bee that's kind of fairly harmless to the bee, but is like in the fat? 'Cause I've heard of, when you eat stuff, what you eat is being stored in your fat. So if they consume something that's distasteful to the bees and the mites, when they eat the fat, they go, don't like that. Um, and thirdly, this is more of an observation question, is this really a race between adaptation between the bees and the mites? Because you did mention how you were saying if the bees could adapt in time to kind of counter the mites, but of course the mites are clearly adapting to pesticides and stuff like that. Thank you.
Yeah, great set of questions. To start with the first one, in terms of the mites, just biology and whether we can sort of utilize that against them. The mites are, well, they're very strange in their biology. The way that their life cycle really works is that they're not colonial. They don't have queens and workers or anything like that. What they do have is that an individual, you have a foundress, and she's the female. And Varroa mites, it's all about really the female mites, because the male and the young mites are all very vulnerable, but the female, she's red and she's got a sclerotized exoskeleton, so she can exist outside of the colony. She can exist anywhere, really. Whereas the male and young ones need to be in a brood cell. So what happens is the female will found a brood or invade a brood cell, usually by piggybacking off of another bee into the open cell while it's tending the larvae. That cell is then sealed, as is a normal process, but then the female is in there, and she will have progeny, and the progeny will include males and females, and then those progeny will begin a sort of incestuous reproduction process where sisters mate with their own brothers, which is interesting because it results in very fast reproductive cycles, and the inbreeding actually seems to increase their ability to mutate and adapt to stress factors. So that's going on inside the cell on the developing brood, usually slowly killing the brood as they feed on it and spread a lot of disease. And then when the brood emerges as an adult bee and the cell is opened, all of these mites that are new females come kind of shooting out into the colony, and the whole process begins again. And so you end up with this kind of exponential mite growth that occurs over time. And I'm sorry, I've forgotten what your second question was.
Oh, no problem. I'm just going to say that's actually incredible. I've never heard of like the inbreeding like that to be an advantage. That's actually quite incredible. The second one was about the fat. Like, could the bees be given anything to make the fat distasteful to these mites if they're eating the fat, and that's a big problem?
Yeah, I think there is some research. I don't know whether it's been published yet, but Samuel Ramsey, who is actually the researcher who discovered that the fat bodies were the the food source for the mites, they were trying to develop some, I can't remember whether it was pesticides or RNA interference, anyway, they were trying to develop agents that the bees could consume harmlessly, but then would become accumulated in their fat bodies, like you mentioned. And then when the Varroa feed on them, it either kills the Varroa or makes them infertile. So there's definitely a line of research that's aiming to sort of leverage that particular aspect and to help try and control the mites. And sorry again, the
That's
first question.
fantastic. And then the last one was only just me, kind of more of an observation. Is this like a race against time about adaptation? Because you were saying before how you were seeing if the bees could survive, just kind of letting it rip with these mites. Could they adapt in time to counter it? But obviously the mites are becoming more adaptive to the pesticides and your strategies faster than the bees are adapting to the mites. That was my just question / observation.
That is something that we've really been bearing in mind from the get-go. So sort of what sounds like you're sort of partly referring to sort of Red Queen hypothesis, where it's the constant evolutionary arms race between parasites or pathogens and their hosts. So what we need to remember in honeybees is that we have this really weird situation where honeybees, like humans to an extent, are really quite decoupled from natural selection, because beekeepers are the ones, looking after them and breeding them. So a colony of honeybees that might die in the wild might survive because a beekeeper treats them, with miticide or gives them extra food. So we have all these honeybee colonies kept by beekeepers that aren't adapting to anything. And then along comes a new invasive parasite, the Varroa, and they're just not ready for it. And they never will adapt because they'll treat them with miticides. So you really have this kind of stalemate where not stalemate, the bees are stagnating, they're not adapting, and the mites will just wreak havoc. So that's where the selective breeding comes in, because we can use that to artificially sort of speed up the process to make already adapted bees. And the final point on that is that we see that the mites, partly we think because of their inbreeding, often develop mutations that basically make them resistant or resilient to chemicals. And the risk with that is that you can often have a very small amount of mutation can suddenly make something ineffective. But because the bees are using a complex behavior to detect and remove the mites, that's a lot harder for the mites to evolve against. And on top of that, because the mites are often getting just eliminated from a colony, you never get enough surviving, to get around the behavior. And in fact, it's worked very well for Asian honeybees, because they still exist, sort of coexist with Varroa mites, because the Varroa mite levels never get high enough in their colonies to destroy them. So they reach a kind of equilibrium.
That's amazing. I've got so many more questions, but thank you very much. I'll yield the floor. Thank you, Doctor.
Z and Krishna, you had questions? Please go ahead.
Hi, this is Z, and actually my questions were just answered, so thank you very much, Thomas. Please come back if you can. Thank you.
Yes. Cheers, Catherine. Yes, I think Jamie kind of asked the question about can they consume something they don't like, because I notice when I go to India, say, I get bitten by mosquitoes, and I'll be in the same space as say somebody else, my other family member that's originally from India, and no one bite, like they don't get bitten at all. And I just find that crazy, because we'll be sharing the same space, maybe even the same bed, and I wake up bitten all over. And it's been said that we could consume Marmite and that would like ward them off because they don't like the taste, the smell, or something. I think it's is it the B12? I think, I can't remember, or B2 or something like that. So yeah, Jamie asked a question, but he said there's a line of research, and that's where my thinking was coming from. And yeah, that's great to know about the whole evolution to bring an equilibrium. I think that's probably the best bit, as you mentioned, why that's not possible at the moment with breeding. And then there's just one more thought process was, well, I don't know whether I agree with this ethically, just from my own kind of point of view, but um, can there be like selectively targeted using some kind of energy beam, like when we use kind of laser on the legs or something, it targets the follicle only and removes it. So could something like that be done to remove the mite?
Yeah, I mean, let me think. There's several ideas that are kind of along those lines. There's a lot of work for mosquito control, which uses these sort of laser mosquito nets. So the mosquito can't fly through this laser mesh because it will be incinerated if it tries to. Although, of course, there's a lot of problems trying to make ones that don't also, hurt people when they set them up. But when it comes to mites, I know of one particular method which I think is already being rolled out, which is a sort of gate with, I think it has like brushes on it. So if a bee is returning or leave it, or especially returning to a hive and it has varroa mite on it, there's this special entrance gate where for the bee to sort of squeeze through, the brushes will remove the mite, and it won't be able to enter the colony. And so I'm sure you probably could produce like a sort of a laser version of that. The only issue is that these are both insects. So like the example you gave of like, to deal with a hair follicle on your leg, right? Well, the problem is the varroa mite and the bee, it's likely that something that physically damages the varroa mite is also going to physically damage the bee. And then there's one final issue that affects, I know, this gate idea, is that the mites do cling on to adult bees to move around, but that's not where most of the mites are at any one time. Most of them are in the brood cells internally in the colony, and it's very hard to access the brood cells without, having the bees do it for you. The amount of technology you'd need to be able to open up the brood cells, and direct something against the mites would be immense. And so that's kind of the difficulty, I think, there.
Along, oh, I'm so sorry. Thank you. Um, because you mentioned the mosquitoes, is there a way to CRISPR to introduce a variety of mites so they cannot reproduce anymore along those lines that what is being tried out right now in Florida and I think in different places in Africa to use CRISPR basically to get rid of the mites?
So you mean the studies where they introduce the sort of infertile, like sort
Yeah,
of
mm-hmm,
Trojan
exactly.
horse?
Yeah.
It might be possible. I mean, in theory, it should be possible if it is with mosquitoes, as far as I know. I think really probably, and again, this might actually be research trying to do this, but I think the difference and why it might not be such a successful option with Aurora is because with mosquito populations, for example, the problem is that you have, this sort of latent mosquito levels that are transmitting disease. And with varroa mites, it's the mites themselves are causing a lot of the problems. So let's say you introduced a population of these non-reproductive mites into some colonies, for example, you'd get an isolated effect in that apiary if it worked, where the mite levels will go down because they're not reproducing. However, those bees are going out and they're getting exposed to mites from other places. So there'd be a kind of replacement because more mites will come in that could reproduce. And on top of that, the problem again with the inbreeding, it's quite likely that if the CRISPR mediated mechanisms aren't using a very complex, large level of, kind of change to genes that it's going to take a long time to deal with, that they'll start to evolve resistance or start to breed it out very quickly because there's this immense selection pressure and very short generation times.
Well, that brings up one little thing. Like, what if this might sound silly, but whatever, I'm just going to say it. What if you bred out like the mite's mouth parts and then they all just died eventually, right?
Well, it would be hard to stabilize that, as in, like, imagine you came up with a mutation in a gene that just this is kind of doesn't really make sense as a concept. But let's say there's a gene that causes the formation of the mouth parts, and if they have a particular allele, they can't have functioning mouth parts. The problem there is that that particular thing is so important to survival that it wouldn't really be able to propagate because they just die as soon as that happens. So you need to be able to somehow spread that covertly into the whole population with no ill effect, and then suddenly it switches on and they all die. I guess that's the difficulty there.
Well, I mean, we could use maybe something like optogenetics or something like that to like trigger it. Like gene expression that's dependent on some sort of trigger, either maybe biting. Is there maybe something that by biting into the I don't know. There might be, but I don't know if it's ethical also to kill all the mites. I'm not sure what we would do to the ecosystem. I think there might be ways to do it, but I don't know how ethical it is. But to give from the audience, Dale Brown, he is in Ukraine and he's asking, due to the impact of the war, the bee ecosystem is quite stressed. Is there a way to optimize to basically save the bee ecosystem using redevelopmental? What is the opportunity to rebuild the ecosystem, and then also in the future to once Ukraine gets opportunity to get rebuilt, to integrate bee ecosystem into the architecture or city design as part of a smart city, if there are planned, like, are there designs out there, or is there any advice or something for that?
I guess in terms of human and bee interactions at the ecosystem level and the threats that bees face, not just honeybees, but really a lot of wild bees and other pollinators, is habitat destruction and human activities like agriculture that displace them or reduce the food availability, those kind of things. So I guess if you were rebuilding a town or a city or any kind of structure in a region like Ukraine, and you were trying to build it back with added benefits for bees and other pollinators, I would say having gardens and planting species of plants that provide food through pollen and nectar, especially, native plants. I think that's something that often gets overlooked. So if you are providing these diverse gardens with lots of native flowers in a small area that you would normally need a large area to produce, that provides these sort of nice little islands of food source for pollinators that in a city could be hard to come by. And so I guess that would be my main recommendation, is just planting plants that are good for pollinators is a really easy thing to do. And it's beneficial for bees, but it's also good for people. It's nice to have sort of beautiful flowers and such in cities.
Thomas, this is Dale. That was my question. So thank you very much. I just want to make sure I articulate one small detail. I'm not in Ukraine. I just support Ukraine. And I'm collecting ideas for rebuilding Ukraine smartly, since there's large parts of it that are just completely demolished. They had to start over again. Also, Ukraine is the breadbasket of Europe. They produce a lot of wheat and also a lot of different types of plants that are in the food system. And I was just thinking about, I don't know if their bee ecosystem there has been damaged by the war. And I was just thinking about, I'm not sure if people who are doing funding to rebuild Ukraine are actually thinking about the pollinator ecosystem and how to revitalize that after the war ends. And also because if you're looking at smart city rebuilding, a lot of times you'll consider personal gardening or other types of green space within a rebuilding, since you're building something new. And I don't know much about bees, but I didn't know if there was a way to actively incorporate some type of bee ecology into that, where the bees and the people could coexist, and you get the benefits of the pollination. So, thanks for your expertise. This is Dale.
Can I just slip in one more cheeky question, Doctor? One thing I'm curious about, you mentioned how the mites can actually mutate faster because of the inbreeding and everything, right? Did they get away with that without like breeding themselves into extinction because like are there simpler organisms or something like that?
Yeah, that's a very good question. Like you say, we normally associate inbreeding as being a very bad thing that creates all kinds of genetic problems over time and deleterious mutations occur. But I think maybe the way to help understand this is that Varroa as a species, that is its method of reproduction. So, whereas for humans and many mammals, inbreeding is not the way that generally we're evolved to reproduce, and it quite quickly leads to deleterious consequences. For Varroa mites, that is it's central to their biology. Now, I don't know exactly how they are then able to avoid the buildup of negative mutations and consequences of inbreeding. I'm sure there is work looking into this, but what I would say is it's clearly there's been very strong selection for Varroa over millennia, as long as they've really existed, against things that cause problems in inbreeding. So if it kind of makes sense from an evolutionary perspective, the very fact that that is how they reproduce and always really have, as far as we know, means that it must somehow be stable, or they must have mechanisms to stop the bad parts of it coming through, so to speak.
That is fascinating. That means that they can truly survive with like a minimal number of them. That's fascinating. Thank you.
Yeah, I think that's because by in animals that are genetically not very diverse, they are species that have cancers that are actually passed on by biting or something, because they are genetically so close, that the cancer can actually be spread like a virus. Um, is there, yeah, that I don't know if that would be another way of, instead of using CRISPR, using cancer, because it takes a little bit to, it takes some time, so there's not this fast reaction, so it had some time to spread around, and one could actually, if this type of cancer wouldn't affect the bees, have the bees spread that cancer, or whatever uh, it would be, to basically kill over time all the mites, because the bees are the ones that travel around, so they would be a quite good carrier of something that would basically slowly kill off the mites.
Yeah, I mean, on that point, I suppose if we're looking at sort of vectors or things that would, spread from mite to mite across the mite population and potentially then act over time to kill them, I think, well, there's probably viruses and other pathogens that actually infect the Varroa because, there's really a virus or a bacteria for almost anything. But we know very little about that as far as I'm aware. In fact, it was only fairly recently that studies that really showed about whether or not bee viruses like deforming virus can replicate in the mite and whether that affects pathogenicity. That was really sort of not that long ago, really an unknown area. So there's the real, I think, conundrum that we face from a scientific perspective is that, like we kind of talked about Varroa biology, it's so alien in many ways. We have no, or at least, again, there's very little information, I think, on what would happen if you even could introduce something like this. Because cancer, for example, or various cancers, I mean, obviously it's an umbrella term, but in something like a Varroa mite that has a rapid life cycle, it would be a balance between, would a cancer ever grow enough to cause it a problem, or would it have reproduced, lived its whole life and died before it ever happened? I mean, in humans and other animals, we do find cancers, quote unquote, that grow so slowly that they'll never kill a person. And that occurs in other animals as well. And I guess we don't know, depending on their genetics, do they even get cancer? Is that a thing that can even happen? So there's a lot of, it's really interesting questions. There's just a lot of unknowns here. And I think that's one of the things that makes Varroa mites so scary, I guess, is that their biology is pretty extreme and very alien from our perspective.
Very interesting. Now I would like to screen Varroa and study their biology, if but
Me too.
I don't
Me
know
too.
it.
They're
Right?
fascinating.
Yeah, they are really fascinating. Interesting. Yeah, I know we are just putting ideas out there, and most of them are very silly, but thank you for having the patience to answering to all of them.
Thank
And
you. Can
yeah,
I
Jamie.
ask one more? It'd be really, really bad to ask one more. This was just about when you were talking about the Pol-line bees and the helium bees that you were breeding and everything. Is this the fact that they're more resistant to the mites? I'm a little bit confused. Like, does this mean that we're only going to have one species of bee then that's going to be resistant, or is polyamory like a term that covers multiple species, but just the type of species that are resistant versus the species that aren't? I don't quite understand how does bees have species? How does that work? I don't. Yeah, sorry.
Yeah, that's a very good question, because I remember when I first started working with bees, I had kind of similar questions about this. Right, so bees, which are honeybees, are in the genus Apis. So that includes European honeybees, Asian honeybees, giant honeybees, which you live in Nepal. You might have perhaps seen some things about those before. They're massive, Apis dorsata. And then there's several other species of honeybees, some smaller, some larger, with different colony sizes. Now, one of those species is the European honeybee, Apis mellifera, and that's the one that generally beekeepers keep in Europe, America, and Asia. Some people keep the Asian honeybee, Apis cerana, but it's less popular because it doesn't produce such large colonies. So it's a single species we have, which are European honeybees, but within that species, there's a lot of different stocks and breeds. So what we can think of that is like dogs. So dogs, is it Canis domestica, maybe? I might be wrong, but let's say dogs as a species are a single species. You have wolves, they're a dog, I think, and then you have all these different breeds of dogs. They're all the same species, but they're just bred to have different characteristics. Some are large, some are small. There's a great deal of diversity, but they all belong to the same species. That's how we can think of different stocks of bees. So they're all European honeybees, Apis mellifera, but some of them are bred to have some characteristics, like maybe they have large colonies. Others might have other characteristics, more or less aggressive. Africanized bees, you've probably heard of those. They're the same species, but they're a sort of ecotype, so like a subgroup within the species that are very adapted to survive well in many of the African regions. And one of the things about them is that they're super aggressive because of elephants. So within the species of European honeybees, we have these different stocks and breeds that are bred for different reasons. So what I guess we're suggesting is that we take the resistant bee stock, and that can be bred with other stocks. You can cross a Cocker Spaniel with a Chihuahua, and you get a kind of mix between the two. You could do that with Pol-line bees. But what it really allows you to do is we could breed in some beneficial genetics to any kind of stock within the same species. I don't know whether that completely covers it, but I think that.
Yeah, that actually does. So you've got the honeybees, but then there'll be the Pol-line honeybees and the non-Pol-line honeybees, and the Pol-line Asian bees and the non-Pol-line Asian. That's what you're talking about, right? And letting them slowly all become resistant to that. That's what I'm understanding you're saying, yeah?
Right. And if you're a beekeeper, you often just buy a particular stock. You buy queens. So let's say I want to start up an operation, I could just buy Pol-line or another stock that's resistant to Varroa, and then I can keep breeding those and propagating them more. I could mix them with other bees, or I could just keep them as they are and see what happens.
Absolutely fascinating. Thank you so much. Thank you so much.
Yeah, I have I'm sorry, but I have another question. You said that the behavior is also very important, right, to become resistant to the mites, right? Uh, I don't want to assume something wrong for the question.
Yeah, that's right.
So can you train the behavior, because having probably a high variety in gene pool for the future is probably important. So maybe we want, I don't know if there's the thought of preventing of just having one subtype of species in the future, if everyone just uses those. Is there a way of training the behavior? Does it, and if, like, is the behavior fully genetic? If the training, does it pass on? So can you create basically a bee culture that gets passed on to the next generation?
So I think what you're asking then is the behavior of mite resistance. So in this case, opening up infested cells and getting rid of the mites. Is that purely genetic, or is it sort of something that they learn, to do it? Can it be kind of trained in and then you pass that on? Well, it is genetic. I mean, we know this because we've identified several of the genes involved in it. And we know that if you take Pol-line bees that have never seen mites before, they instantly know what to do. But the reason it's genetic is that it's not the behavior that's being coded for in their genes. Actually, the genetics is about them just being able to detect the mites. Because the problem that most of the European honeybees have is the mites are there and they're being eaten alive, but they don't even know that they're there, because it seems anyway, it's likely the mites copy the hydrocarbon profile of the bees, which they use for recognition. And so they don't know the mites are there, even though it's obvious. So what Pol-line and many other resistant stocks do is that they seem to be able to respond to various cues that suggest that mites are present. In the case of Pol-line bees, we're not yet 100% sure, but we think that they're responding to the brood producing some kind of chemical signal that says, hey, I'm infested. And then the worker bees, as anyone really would, the minute they know there's a problem, they'll act upon it. And then that, of course, can be passed through. The ability to detect that is passed genetically. And so you can continue to propagate it over time.
It did say in your paper that the Varroa was associated with different pathologies and behavioural change in the bees, actually. So I was actually wondering what behavioural changes did they cause in the bees?
Yeah, so there's a lot of things, but I suppose some of the interesting things are that if Varroa infest brood and transmit deformed wing virus, so there are various strains of it, but if they transmit enough deformed wing virus, what happens is when the brood emerge, their wings are completely crippled and they're alive, but they can't, do the job of a worker bee. And so they end up being a kind of just burden. Eventually they'll die, but often you have colonies where many of the workers are sort of incapacitated to a point that they can't do their job, but they still require food. And that level of sort of behavioral change in terms of the composition of the colony could be pretty bad and can sort of build up over time. On top of that, you do have various other behaviors that the bees will use to respond to Varroa mites, because sometimes they, to an extent, are able to detect signs. But overall, the key problems we're really facing are proliferation of disease and inability to detoxify other environmental inputs that they're getting because of all the sort of fat body or damage to the workers.
Katerina, are we going to let the doctor go in a minute before we actually ask him more?
Yes,
I could do this all
agree.
day. I
I
could do this
know.
all day.
I'm so sorry for asking all these questions, and probably most of them are quite, coming from ignorance about bees and their ecosystem. So I really appreciate the patience you have answering our questions, and thank you so much for your presentation and for your work. And I wish you all the funding and all the luck
And
that you
low
can have.
bureaucracy.
We are cheering you on. Please never give up on the bees, and I hope the world is treating your research and yourself right, so you don't give up on it ever. And
Definitely, this is so good. Thank you so much. This has been absolutely fascinating, Doctor. Absolutely fascinating.
It's been a pleasure to talk on it. It's always nice to have some good questions, because a lot of the time they're things that I wouldn't necessarily think about, and then when someone asks it, you kind of think, oh yeah, do we actually know the answer to that? Is that worth investigating? But yeah, it's been really fun, and a nice different format as well, just having the audio only, quite a pure format to do it. But so I'm probably gonna head off to the pub now, so it was a pleasure talking to you.
oh, enjoy.
That sounds good. Have one for us, and we hope you come back sometime soon, because we'll probably, when you find anything else, please come back. We'll have more questions for you.
Yep, exactly.
Great.
So
Okay.
yeah, enjoy your weekend. Happy
You too.
Friday, everyone.
Happy Friday.
Have a good
Bye.
weekend, Doctor.
Everyone.
Bye, everybody.
So much.
Thank you, everyone. Nice to see you.
Plant
Thank
more.
you, Dale. And thank you, Z, for the picture, for changing it to the bee.
Oh, well, I will say, look, I caught this picture yesterday. I've been looking to take a picture of the bumblebee eating off of the black gum Nissa sylvatica wild tupelo tree, and I caught it yesterday after 5 years. So this is amazing. Thomas, thank you. I love you all dearly, and I'm just going to shut my mic off now. Thank you. Meow.
Oh, yeah, that's wonderful. Thank you. What a coincidence. That's perfect. Yeah, thank you everyone for asking questions. And we have a room at 5:30 with Krishna. You met her, she and Faris. He's a editor for a scientific journal. They will give some advice how to write a scientific article. So if you're interested in that, tips, advice, and how to do that, well, come back at 5:30 p.m. EST. Yeah, and thank you so much. Enjoy your weekend. Yeah, I hope we'll have amazing happy bees in the future, thanks to Thomas. So thank you, Thomas, and bye, everyone. I'll close the room in 3, 2,
Thank you,
1.
everyone. Be
Bye.
good.
Thank you.
Bye.
Bye.